Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Welcome to the series of lectures on food microbiology. This is the second lecture in the part where we are talking about the spoilage of food. In the first lecture of this series, I said that depending upon the nature of the food, whether it is a cereal or cereal product, whether it is a vegetable or a fruit product, whether it is milk and milk products, or whether it is a meat, fish or poultry product, the type of damage or spoilage caused to the food and the type of microbes which cause the damage are different. To recap on what was covered in the last lecture, let's see some important microbes which, for especially the molds, which cause post-harvest disease of the fruits which are harvested. You have the Alternaria. As I already said, the mold or the infection is normally caused as a rot disease, and depending upon the type, the visual color of the type of seed which is seen on the surface of the fruit, you call it as a grey rot or a soft rot or a red rot, depending upon that. So, Alternaria causes brown to black spots on apples, stone fruits and figs. It also causes a stem end and black rot of citrus fruits. Then you have the Aspergillus species. This Aspergillus, this causes black rot on the peaches, the nectarines, the apricots, citrus fruits and the figs. But you have the Botrytis diplodia. This causes what we call as the cushion or the crown rot of bananas, the ripe rot of pawpaws. Botrytis, it causes the grey mold. See, in the first case we talked about Alternaria causing a brown or a black spot. Here in the case of botrytis, we are talking as grey mold rot of apple, pears, raspberries, strawberries, etc. Cladosporium, this is a restricted rot with a black core on stone fruits. This shows also in the case of olive, it is a olive green growth on raspberries. It causes the black rot on the grapes. So, depending, Cladosporium can cause very type of visual changes depending upon the type of fruit we are talking about. For example, you see it is on the grapes, it is a black rot, whereas in the case of a stone fruit, it is a grey black core on a stone fruit. The Chalilotrichum causes brown to black spots on citrus fruits, mangoes, avocados, etc. The Diplodia causes stem end rot of the citrus fruit, avocados, mangoes, and it also causes the watery tang brown root in the case of peaches. Fusarium, it causes the brown root of the citrus fruit and pineapples. Geotrichum causes the sour root of citrus fruits. Gliosporium causes the anthracnose, and Menilinia causes the brown rot of stone fruits. Mucor, this causes the soft rot of strawberries. The Nigrospora causes the soft watery rot, that is in the bananas, meaning it is a squishy stuff. So, the moment you take the fruit and slightly press it, the water, you will see water coming out of the banana. So, the nature here is different. Then Penicillium, this causes a green and blue mold rot of citrus fruits, the blue mold rot in the apples, grapes, pears, etc. In Phomopsis, you are going to see the stem end rot of the citrus fruits. Phytophthora causes the brown rot of apples and citrus fruits, the leathery rot in the case of strawberries, meaning the outer nature of the strawberry fruit becomes leathery, tough in texture. Rhizopus causes watery soft rot of apples, pears, stone fruits, etc. Trichoderma causes the cocoa brown to green rot in citrus fruits. So, the commonality what you see in all these damages or spoilages, you are seeing a change in the texture, you are seeing a change in the color on the outer epidermis of the fruit concerned, as well as in some cases the nature you will see, as we talked about, you are skirting of water from the fruit, a change in the structure, damage, visual evidence of the damage because of the fungal infestation would be evident during the post-harvest storage of the fruit concerned. So, all these are indicators that there is some problem with where you are during your transportation and storage, and the fruit has got infection during the particular process. Now, the next type of plant product which we are going to talk about are the cereals, the grains. The cereals belong to Graminae or the grass family, and these are the major sources of carbohydrates in human diet. The microbiology of the cereals during growth, harvest and storage is dominated by the moles, and it is convenient to consider two groups of fungi here. As we talk about cereals, we have to start talking about the fungal infestation straight from the time the seed germinates. As I said, as the plant grows, the outer epidermis of the stem is actually very thin. You will have the stem boring, the cell sap sucking insects boring and making holes, and the moment a fungal spore enters, it is going to start germinating inside, and once a plant gets a fungal infection, because all the plants have interconnection, the what we call as the xylem and the phloem connection, once the plant gets infected, the infection will spread throughout the plant. So, it is there inside. You might or might not have the visual evidence as a rot or a discoloration in the type of the leaf or the stem, spots happening on the surface of the leaves, but even without that visual indicator, sometimes the plant might be infested. The fungal hyphae would spread throughout inside. Okay, so, when the cereal is when you harvest the grain and store, inside it also you might have the spores concerned, which might germinate if your storage conditions are not actually proper. So, in this case, and the second aspect which you have to think about is the most of the grains which we harvest during physiological maturity, they have a high water content, and before we store them, we reduce, the AW becomes reduced, that then only we are able to store the grains. If at any point during the storage you are going to have a change in the relative humidity and the temperature of the storage area, this will cause a disruption in the water content. And you always have to remember, the seeds, even when dehydrated, are living, they respire. So, these respiring seeds, the products of metabolic products, byproducts of respiration, as you know, is carbon dioxide and moisture. So, if sufficient aeration is not there, this moisture will accumulate. This in turn will cause microbial growth, causing damage to the stored grains. So, that is one aspect. Fusarium culmorum and Fusarium graminearium causes both stem rot and head blight of wheat and barley in the field. This we are talking about the infection in the field even before harvest, and these field infections may lead to more extensive post-harvest spoilage if they are stored at a too high water activity. So, this was what we were trying to talk about, whether it is rice, wheat, barley or any type of grain concerned. If the mother plant was infested during in the field, post-harvest, the spores would be present inside the grains which are stored. So, if they are stored at a very high humidity or not in a temperature or not under conducive conditions, the infestation still can happen from within, causing damage and loss of the grains concerned. The so-called storage fungi are well adapted to the constant conditions of the cereals in storage, and generally these can also grow at low water activity. This picture actually shows in the field the wheat rust. This is the ergot fungus which is infesting that. Now, the most important genera of storage fungi are Penicillium and Aspergillus. Although species of Fusarium may also be involved in spoilage when the grain is stored under moist conditions. Grains and seeds normally have 10 to 12% moisture, and this lowers the AW and thus will inhibit the microbial growth. In cereal grains, during harvesting, processing and storage, if AW increases beyond or above 0.6, molds can grow up. So, when cereals or grains are stored, the most important point to talk about or to take care about is what is the water activity there. How it is the dehydration level, what is the water activity. It should be even less, where even the most tolerant fungi cannot grow. Many types of lentils, beans, seeds are germinated and we use them as ready to eat products. So, here also when it is a ready to eat product, see, you are already germinating it. So, germination will make available the nutrients which are present. If the nutrients are available, it is available for the microbes also, not only for humans. So, that is an ideal nutrition medium for the microbes to grow. So, there the usage as well as storage of these ready to eat foods have to be specifically taken care of. The storage condition of these how we are storing, what is the temperature, what is the water activity. This becomes very important, especially when we are going to talk about ready to eat foods. And during storage, gram positive and gram negative bacteria, yeast and mold can grow and produce off flavor. So, how do you know that you are going to have mold growth or you are seeing mold growth on the cereal? First is you have a reduction in the germinability of the grain. That is, if you put the seeds, the grains are actually seeds. So, normally when they contain the embryo. So, if you are going to put it for germination, after you will see sprouting. So, when you are having an internal attack, the germination capacity of the seed will reduce. So, that is the first indicator. The second indicator is discoloration. The change, you will see a visual change in the color of the grain. Third, the production of mold metabolite including mycotoxins. Then, fourth would be a demonstrable increase in temperature, meaning in the storage area or in the storage bin or the vessel where you are storing the grains, the temperature of that will rise up. The culprit here is respiration of the seeds, because as the seeds respire, it produces a little bit of heat. Okay, and then you are having an increased amount of carbon dioxide and moisture. This is, and you are keeping the bin tightly shut. So, the moisture will condense and then fall back about, and when this will increase the water activity. This will cause the germs to sprout, the fungal spores will germinate. So, this becomes a cycle causing spoilage of the stored grain. You see the production of musty odors, caking. They will become stuck together as a mass instead of the individual grains becoming seeing as ideal single. They will become caked together to form a ball-like structure. And finally, a rapid increase in the water activity, leading to finally complete decay with the growth of a wide range of microorganisms. Initially, the visual evidence would be less, you might not notice it, but the trigger would be offered by the growth of one type of microorganism, would in turn promote all the other microbes which are spoilage organisms to come and attack it, and all of them would proliferate. So, ultimately you will see a wide array of microorganisms combining together to spoil the particular product concerned. High moisture cereals like barley provide a highly nutritious winter feed to the cattle. So, for this, long-term storage of such material can be achieved by lactic acid fermentation or by careful addition of fatty acids like propionic acid. So, in temperate areas where you have well marked winter and summer and where the temperatures go below sub-zero, getting fresh fodder for cattle would be difficult. So, there we keep high moisture cereals like barley as a grain. Okay. So, when you are going to store these grains for a long time, to prevent them from getting degraded or getting spoiled by microbes, we do lactic acid fermentation there. Okay. But in spite of this, even with this fermentation, you might have spoilage because aspergillus flavus can grow in it to some extent. Now, but because of this fermentation by lactic acid fermentation, the aspergillus might not have proliferate well, but the problem here is it is still growing. and aspergillus produces aflatoxins. Okay, so aflatoxin B1 is the one which gets produced under these conditions. And when this barley containing aflatoxin B1 is fed to dairy cattle, there would be a possibility of aflatoxin M1 being secreted in the milk. Then it becomes a problem for human food and not a problem of animal feed. So what will happen is that the human being is not directly consuming the barley which has aflatoxin B1, but because it was fed to dairy cattle and the humans are consuming the milk and the milk products from that cattle which consumed grain with aflatoxin B1, it is synthesizing aflatoxin M1 that was secreted in the milk, and that is what is getting consumed. So now this becomes a contamination problem in the human food concerned. So this shows actually damage to the rice seeds. You are seeing the fungal growth, the black spots. This is actually fed to poultry. So if you see such widespread damage in the corn which is fed to the poultry, naturally this is contaminated food which cannot be used. Cereals, flour and dough products. When you are using rice flour or wheat flour for making a variety of products which are fermented, non-fermented, it can be the idli dosa of your South Indian food, or it can be the bread, pasta, pizza, whichever, you are going to use the flour of the cereal. Now, the flour which has been milled still can contain microbial contamination. They will have the same microbiota which has been passed from the soil through the original parent plant to the storage area and during packing. But because normally the water content is less, the flour will not show microbial growth. But still, if after milling, if the storage process and the temperature gets changed, it can proliferate microbial growth. The second aspect in this case would be the bleaching agents which use the food. When the dough and the dough products and the flours which get bleached to have an acceptable color to the consumer, the chemicals which are present in the bleaching agents can prevent the spoilage organisms from growing there and thereby causing damage to it. So, when we talk about plant and plant products, in a broad sense, we categorize them into two areas. We are talking about cereals and the flour, where the storage, if it is storage as a cereal, the water activity is less. So, there you will have more amount of fungal germination and spoilage there when the water activity, in spite of decreased water activity causing contamination. So, the type of precautions you take for the cereal and the dough flour products are totally different, whereas the other type of plant products, your vegetables and fruits, they have a high possibility of getting damaged by the microbes, basically because their water content is very high. The nature of their nutrient composition is totally different. But in all these cases, the commonality is whenever you have a microbe infecting them, naturally there is some sort of spoilage which in turn can be evidenced by either a change in the smell, flavor, aroma, or even a nature of the fruit concerned or the texture of the fruit concerned. In all these cases, we have various preservation methods like drying, heating, freezing, the different types of processings, the different type of additives we add, the chemical preservatives. All these to some extent will prevent the growth of the microbes, thereby reduce the spoilage as well reduce the microbial load of the food concerned. This in turn will cause an increase in the shelf life of the product concerned. So this is one part where we have discussed about the plant and the plant products. We are talking about microbial spoilage to plant and plant products. We were discussing about the nature of the damage caused by microbes to different fruits and vegetables, and even in many cases what is the type of damage seen to the food material. If it is meat and confectionery, if you see a slimy layer on the outer side, this is because of a polysaccharide formation. This polysaccharide formation is because the meat has been infested by Pseudomonas, leuconostoc or Bacillus species. If you see a ropiness in bread and milk, this ropiness is because of the polysaccharide formation caused by alkylaginous species. The milk can have a bitterness because of Bacillus cereus. Cheese, you will see holes. These holes are produced because of the escape of gases which are formed inside. This gas production is because of coliforms. You will see softening or rotting in fruits and vegetables. This is because of the enzymes pectinases, cellulases, xylanases caused or produced by Erwinia, Clostridia, etc. Milk and meat can become curdled by acid production by lactic acid bacteria. The visual evidence of spoilage is, if it is gas formation, you will see holes, bubbles, fish eyes, etc. normally seen in cheeses, olives, cucumbers. Basically, the organisms which cause them are coliforms, yeast and the lactic acid bacteria. If on the surface, if you see growth, it will be moles. Many fruits like potato salad, beetroot, etc. caused by fungi, pichia, etc. Discoloration is normally seen, for example, beetroot. Beverages and brines can have cloudiness or turbidity. This is caused by yeast formation. Then you see discoloration as an indication, the red spot, browning, blackening, change in the color. For example, meats and egg, you will see either fluorescent color or a pink color in sauerkraut, red spot in cheese, browning in brine vegetables. All of these changes are visual evidence that you are seeing microbial growth in them. The microbes which normally grow in specific food are like either Pseudomonas, Lactobacillus species, Pseudomonas, etc. All of these actually changes. Their metabolites will discolor and start producing the browning or blackening or greening. To summarize, in this lecture we saw what are the types of damages which normally happen to plant and plant products, whether it is a vegetable, fruit or a drink or a beverage, or whether it is a cereal or dough. In all these cases, depending upon the water activity and the nature of the food concerned, you saw different types of microbes growing up, and depending upon the nature of the microbe which grew up, the visual evidence as well as the biochemical parameters of spoilage in each of the food product became different. Thank you.